Good evening, everybody. So my name is Maria Antonietta Loi. I'm a member of the board of the KNG, and I'm also professor of applied physics at the University of Groningen. And it's a great pleasure for me today to introduce one of my dearest colleagues in applied physics. So Elisabetta Chicca studied in Rome at the University of La Sapienza, physics, where she did a master thesis. I think she was one of the first people studying how to use a silicon circuit for simulating spiking neural network. And then she moved to Zurich. She did a PhD between ETH, the University of Zurich. And actually, she had a PhD in two different topics, in, let's say, more physics and engineering of neural networks, but also in neuroscience. So she's a very multidisciplinary person, that she combines, let's say, biological understanding of the brain and of the brain of insect, for example, with the simulation of the behavior of the brain in electronic circuits. So after the PhD, she stayed in the same institution as a group leader for several years. And in 2011, she moved as a professor in Germany at the University of Bielefeld. And in 2020, we were very lucky to steal her from the University of Bielefeld and to bring her to Groningen, actually as a professor in the KNG-Groningen Institute that was at that time founded to study neuromorphic electronics and neuromorphic new type of computing. So by then, she has made a very successful group in Groningen, with many younger people very active in this field. She has acquired an enormous amount of money, grants, ERC synergy, and today we are very happy to listen to her and to the new field that will explode soon, that is neuromorphic computing.
Thank you, Maria, for the very nice introduction, and thank you to the KNG for inviting me. It's really a pleasure to be here in front of this audience. And yeah, the talk is about inside the insect brain, as Maria already anticipated, how tiny minds navigate a complex world. But before I move into this topic, you have heard already some weird words like neuromorphic. And I wanted to start from there. What is neuromorphic engineering? So this is a term that was coined by Carver-Mead in the late 80s to describe integrated circuits, specifically CMOS technology, the same technology that is used to build your computers and your cell phones, and to use this technology to mimic the computation observed in the nervous system. And there are two important motivations. I believe most probably at that time there was only the first one. And the first one was understanding biology with a concept that is also inspired by what Feynman wrote on his blackboard some days before his death. So there is a famous picture of his blackboard in his office where there is this sentence, "What I cannot create, I do not understand." And that's a principle that is very close to people like me doing this kind of research, in which we try to understand the brain by building systems that operate with the same principles. And then a second motivation that is quite hot these days is then using this system to develop new technologies. And before I say much more about what Carver-Mead was proposing and his ideas about how to understand the brain by building the system, I would like to play a video. And I'm telling you that this interview that he gave is from 1991, so a long time ago. And let's see what he says.
Time. We have enough technology to build any...
Let me try it again, because the audio wasn't there yet.
I think at the present time, we have enough technology to build anything we could imagine. Our problem is we don't know what to imagine. We don't understand enough about how the nervous system computes. to really make more complete thinking systems. We have little tiny insights that we've gotten from studying biological systems. And we can build little systems based on those, and we can make them work, and we can understand a little more, and then we can go and ask the biology some more questions, and we can learn a little more. But it's slow going. The brain is an enormously complicated thing. Even very small parts of it are way beyond anything we understand. So we're really limited by our understanding, not by our ability to build things.
So I think this is even more true nowadays. Many years past, technology made huge improvements. Neuroscience as well, but I think the gap became bigger. It didn't become smaller, because what we can do nowadays with technology is amazing, but we are still lacking the understanding. And so this going back and forth that he was talking about, this is the kind of research that we do in my group. The group is called Bio-Inspired Circuits and Systems. And this is still what we are doing, and that's the topic of today's talk. One question you might ask, because nowadays you see a lot of artificial intelligence, you see a lot of so-called intelligent systems, and you might ask, why am I doing the research the way I do it? So by using integrated circuit, what he proposed in the late 80s. So what is the motivation for keeping up this tradition, especially if you consider that this is an approach that is very costly? So designing those full custom chips, it's much more difficult, especially with a small team, than designing a new computer, because we don't have all the automated tools that are used in designing a new computer. We do a lot of things by hand. That's why it's called full custom CMOS. So what is the motivation? One thing to consider is what you see in this plot. So here on the x-axis you have clock frequency, and here there is a diagonal line where you have years starting from 1971. And what you see here, all these dots are different processors that were developed over the years. And over the years, what you see is that the clock frequency have been increased further and further. Because of course, we always wanted faster computer, because we want to do more and more computation in a very short time. And that's also why current AI can do what it can do, thanks to the fact that the clock frequency has been increasing in this way. And at the same time, what happens as a side effect is that whenever we increase the clock frequency, we also increase the power density. So the faster these computers are, the more energy is consumed per square centimeter. So if you look at this graph, what people from IBM have done here is they've put a star here in this corner, and it's labeled brain. So of course it is arguable if these exact numbers are correct, but it's a reasonable estimate. And so it's telling us that our brain does for sure something different. So it operates at a very low clock frequency, and it consumes very little power. So to give you a concrete idea of what that means, if we think about AlphaGo, which was the first computer program to defeat the world champion in the game of Go, it consumes 10,000 more energy than a human sitting at the same game table. And you should not forget that AlphaGo is just playing the game of Go, while the human can perform so many tasks that an artificial machine is not able to do. So there must be some fundamental differences that we need to understand. And we understand some of them. For example, if you look at a computer, you have this kind of architecture where you have a processing unit here, and you have a memory here. And there is a communication channel between the processing unit and the memory. And then again, something quite old, an old quote by John Bacchus, 1978. Programming is basically planning and detailing the enormous traffic of words through the von Neumann bottleneck. The von Neumann bottleneck is exactly this communication channel I was talking about. And much of that traffic concerns not significant data itself, but where to find it. So already in 78, it was clear that the computer was spending most of the time bringing data back and forth in this channel. And again, with all the years that went through, situation didn't improve. CPU made enormous progress. The communication channel improved, but not as fast. So the gap is increasing exponentially over time. So this is to give you the background about the field and about some reasons why we believe that using a computer to simulate cognition, to simulate an intelligent behavior, it's probably not a good idea, because the physical substrate is fundamentally different. And that's why we have still high motivation to simulate what we study, to emulate what we study on a physical hardware instead of simulating it. So now I think we can start with the topic of the talk. And that's, again, the title I read before. And I want to start with another video. This video is about recent research to try to understand some fundamental principles behind insect fly. So how insect decides where to go when they fly. So let me play the video.
Flies make life or death decisions in the blink of an eye. Their rapid turns, called saccades, allow them to dodge predators and obstacles with incredible precision. At the center of this behavior is a specific neuron, DNP03. This descending neuron connects visual input from the eyes to the motor systems that control wing movement. When DNP03 detects an object approaching from the same side as its dendrites, it springs into action, sending signals to help the fly decide whether to turn. But here's the fascinating part. DNP03 reacts to looming stimuli both during flight and at rest. However, during rest, it doesn't fire action potentials. This suggests that while DNP03 is responsive in both states, its full activation, and thus its role in triggering a turn, is specific to flight. Another key discovery, DNP03 doesn't just react to a stimulus, it stays active even after the stimulus is gone. This sustained activity strongly predicts whether the fly will execute a saccade. To confirm its role, researchers used optogenetics to artificially activate DNP03 in free-flying flies. The result? The flies reliably executed saccades, proving this neuron's crucial role in flight control. But DNP03 isn't the whole story. The variability in saccades suggests that other neurons and circuits work alongside it to fine-tune the fly's decisions. Understanding this complexity helps us uncover broader principles of how animals balance sensory input and internal states to make decisions. This research not only unravels the neural secrets of flight in fruit flies, but also paves the way for understanding decision-making in other animals, even designing smarter robots that mimic these behaviors. To learn more about how tiny brains make big decisions, check out the full study. Stay tuned for more research news from the Neurobiology of Flight Control Group at the Max Planck Institute for Neurobiology of Behavior.
So our neuroscientist colleagues are studying flight in insects in the way you have seen presented in this video. And today I'm going to tell you how we do a complementary kind of study, often in collaboration with the neuroscientists, to try to help them figure out even more and to try to guide their experiments given what we discover in our experiments that you will see later. But I'm going to build this up slowly by introducing the fundamental concept one by one. And so we will start with event-driven sensing. In particular, we will look at event-driven vision. But what I'm going to say is relevant for all sensing. So if you look at how sensing is done in artificial system, we have what is called clock-driven sensing. And I'm going to explain you what that means. So if you think about vision, imagine that this is the luminance over time that a pixel is experiencing, a pixel in a traditional camera. So what happens in the traditional camera is that at some point it was decided how fast the camera should acquire images. And that's defined here by these vertical bars. So this is the clock. At every time there is a clock cycle, a data point is going to be acquired. And so in the case of a camera, this would be the luminance of a specific pixel. But because it's a camera, there is a full image. So at every clock cycle, a full image is acquired. And this is good and nice as long as the signal is changing with a dynamic that is comparable with the time of a single clock cycle. But now what happens when the signal, for example, stops changing? So when nothing changes in the visual scene, what happens is that we are going to acquire the same data again and again. So you see that we have what we call redundancy. So at every clock cycle, I'm acquiring the same value of the absolute luminance. And then I generate a lot of data that is mostly useless. And then a processor will need to process it somehow. So it's kind of a waste, in, both in terms of the data generated and the processing needed to figure out what it means. Then imagine also that the signal could change much faster than the clock cycle. And that's what you see here. Here you're going to have information lost because the clock cycle is fixed. It's not going to adapt to the change of the signal. In nature, things are done differently. And most of our sensing are especially receptive to changes over time. So if you want to formalize this kind of sensing, it's something that you can call event-driven sensing. So now this is the same plot of before. You have the same signal. You still have time here, but now you're not discretizing time with your clock cycle. You are instead making another decision. You are deciding how precise you want to be into measuring your variable, in this case, luminance on a pixel. And so in the first part of the curve, you're going to get something very similar to what we had before. But now, when the signal stops changing, we don't produce useless data because this value, it's still valid for a long time. And now, at the same time, when the signal is going to change fast, in a fast way, we are going to adapt how fast we acquire just because of having these thresholds for acquisitions. And then you see that you will have quite some data here that lets you capture the dynamic of your signal. So in that way, we are adapting the acquisition of data to what the signal is doing. And you should notice that while here I had long arrays of numbers, here I have just one number. Because now, while before, the clock cycle was one clock cycle for the whole visual field. So, and that's what you hear being named as frame rate when you buy a television, for example. So it's how fast this clock is going and how fast you're showing or acquiring imaging in case of a camera. Here, because you have this adaptation, of course, you're going to adapt locally. Every pixel is going to decide when to acquire data. So you save twice in case of before the redundancy, because before you were acquiring a full frame, even if nothing was moving, and now you acquire nothing if nothing is moving, and you acquire only where needed if there is a change in signal. And so this is all kind of abstract, and that's why I brought some hardware here, because, yeah, I can show you a picture, and this is a picture of the kind of data you get from such a camera, but it's still not intuitive enough to see this static picture. That's why we have here cameras, and Vincent is please gonna come and make sure they work, and so we can go on with the first demo. And of course, there's something called demo effect, which is you prepare a demo, you test it many times, then you arrive to the location, you start everything, you make sure that everything is working, and then it's there peacefully working until you need it again and it stops working, but you can revive it. So, do I have a pointer here? So here you see these events over time. So the events are generated here, and then they travel over time to show you what happens. And so these dots that you see are the responses of pixels to changes. So if Vincent is not walking in front of the camera and everything is still here, the camera only produces some noise. So it's not a perfect system. There is some noise. But otherwise, you see edges. And you always see two edges, because if I move something dark in front of something light, I'm going to generate a change of luminance at the edge of what I'm moving. So when I move in this direction, if this is darker than the background, at this edge, I'm going to generate events that are called off events, because the pixel is going to see a change from bright to dark. On the other side of my finger, I'm going to see on events, because the pixel is now going to see a change from dark to bright. And so that's why also in the picture that I had before, there was black and white, and also here you see that you get two different colors at the two different edges, and they switch position when I invert the direction of the motion. And during the break, we will keep this hardware here running, so you will be very welcome to come and play with them, because the focus is quite close to the cameras. So from the audience, there were some people trying to wave, but you see a little bit. Maybe if you can do it again. Yeah, but the focus is somewhere closer. That's why you don't see much over there. OK, then we can switch back to the slides. So now the next concept, optic flow. As you have seen from this camera, you were seeing things that are moving. And optic flow is something that we perceive quite often, for example, as in this image when we are on a train. And it is quite instrumental to understanding visual scenes. And it is quite well known that insects use this. So what is optic flow? Optic flow is the apparent motion of objects in our visual field caused by the fact that we are moving. So if I'm on a train, I'm moving, and so I see things moving. That's called optic flow or motion flow. So how do I measure it? I want to measure how fast the optic flow is. So what I can do is I can take two pixels on a camera, and I can think, OK, if I want to measure the velocity of an object moving from this pixel to this pixel, I can measure time. I can measure the so-called time of flight. So if the time is very short, it means that I have very high motion. If the time is very long, it means that the object is moving very slowly. So we proposed a computational element to measure time between two events. And as I said at the beginning, these kind of things are valid for vision when I talked about event-based vision, but they are valid for all senses. And so in all senses, a lot of information is carried out by the so-called spatiotemporal features of the signal. So how the signal changes over time and in space. So this computational element that we designed to measure time differences between two events is quite powerful, and we have been applying it to all sensory, to almost all human sensory modalities, everything except taste. But today I'm going to focus on vision. So how does it work? Imagine that you have two channels, two input channels. And in these channels, event can occur, and we call these event spikes, and here they are represented by this tiny vertical bar. When one spike occurs on this channel, we do something that we call sampling the time. So basically what we do is changing the value of a variable. So this variable was, for example, 0 at the beginning, and then when this spike occurs, we change its value. And then we let it decay over time. Now, if there is another spike on the other channel, early enough to have a value different from 0 here, we use that value to modulate the gain of a synapse. So a synapse is a connection between two neurons, and neurons operate with electrical signal, so a synapse is normally producing a current that is going to be integrated by the sum of the neuron. And so let's see how this works. Let's start from here. We name those two channels facilitatory and trigger. And I always confuse the name, but one way to think is that the trigger is triggering the measurement of the time. So let's start from there. I have a facilitatory spike first, and that generates the change in the variable that I was describing you before. And then over time, the variable decays. But now, when the trigger spike comes, I sample the value of this variable, and I use it to decide the amplitude of this small signal here. So the amplitude of that signal is modulated by this value, and because a lot of time passed, this value is very small, so the amplitude is small. And this signal now is a current that is sent to the neuron, and the neuron is an integrator, so it's summing up the current as it comes. So as you see, this is the signal that you see in the neuron after the integration. And so it's building up because there is current, but then the current goes down, so everything goes down. And in this case, the neuron didn't reach a threshold. The neuron is an integrator with a threshold. And what you see here is that if the neuron reaches the threshold, then it's also producing spikes, events. So the green bars now are the output spikes of this neuron. So what happened here is that the two events were closer in time. So when the second one arrived, the variable was big enough such that the amplitude of this current was big enough such that the neuron could reach threshold, and in this case, it could produce two spikes. And now you can easily understand that the more the two events are closer in time, the more spikes I'm going to see at the output of the neuron. And you're also going to understand that, of course, if the order is reversed, then I see no spike at all, because this is the time in which I trigger, and now the variable is 0, so there is no current and there is no output spikes. So now you can measure how this computational element encodes time by doing many of those experiments in which you change this delta t, the time difference, and, for example, you count how many spikes are produced by the neuron. And that's what we have done here. I'm not going to talk about the integrator circuit that we designed, but I wanted to tell you that this data is from a chip that we designed here. So this is a silicon circuit, and the data that you see is measurement from the silicon. And so here you see the same kind of experiments that we have seen before. And the idea is that you do this many times with different delta t's. And then you can quantify, for example, the number of spikes that are produced as a function of the delta t at the input. And you see a very nice encoding of the delta t in terms of number of spikes. At the same time, you could look at the time to first spike. So if you see here, after the trigger spike, it takes some time for the neuron to integrate the first spike. But of course, this time is much shorter if the delay is short, because if the delay is short, there is a lot of current to be integrated. So this is plotted here, time to first spike as a function of the delta t. And you might notice something really nice here. You are trying to measure time, and you want to have possibly a good resolution. You want to be able to distinguish, for example, I don't know, 25 milliseconds from 28 milliseconds. And maybe you want to distinguish 2 milliseconds from 4 milliseconds. And if you look at these two graphs, you will realize that the two quantity, the number of spikes and the time to first spike, give you their best resolution in very different regions. So, for example, the time to first spike in this region doesn't tell you much, except that the time difference was very small. So this is flat. So if I change the delta t, my measurement doesn't change much, which means I cannot distinguish two different delta t's. I can only say it's very fast. But instead here, for the small delta t, I have a very nice variation of my measurement. So by looking at the number of spikes, I can measure well short delta t, which I cannot do for long delta t with the number of spikes. But on the contrary, here I have a very nice resolution, because for a small change in delta t, I see a big change in the number of spikes, in the time to first spike. So it's a very nice combination. In one system, I make sure that I have a high resolution overall, even though each of them has only a resolution in a given region of my variables. So let's go back to optic flow. Now, if you bring what we told before about how to measure optic flow, you want to measure the time difference between an object generating data at this pixel and then later on generating data at this other pixel. Now you can connect the pixel to a time difference encoder. So you say, okay, now this is my facilitatory channel, and this is my trigger channel. And so I'm going to be sensitive to motion in this direction, and I'm going to have a TDE that can tell me how strong is the motion in this particular region of my visual field. And so if I have an arbitrary moving object, like a bar, moving in this direction, I can experience the motion in the x direction by placing a TDE between these two pixels. And then, of course, I can decide to do more. I can decide to look, for example, at the 4 cardinal directions, and then use this to build an understanding of the motion flow in my visual field. And that's what we have done in this work, in which we use an event-based camera, and we use our TDE model to compute the local optic flow at every location of the visual field. And so we have for each pixel, we look at the up direction, the down direction, the right and the left. And then we build these maps of optic flow. And then we bring them together. And so we are able to construct an image that is now telling us how strong is the motion flow locally. But we can also change how much we integrate this concept in the space, such that we can look at more and more global flow. And this brings us to the second demo, where you will see another vision sensor with this algorithm attached to it and performed on the computer. So before, the computer was only showing you data from the camera. Now it will show you data from the camera, but also the outcome of this motion flow algorithm. And this is the second demo, and it's planned to be right after the break. So as I said before, there will be the possibility to play with it if we don't have the demo effect again. Demo effect. It's 100% probability. 2/2. Maybe. No? You got it? Okay, this is just an introduction to the break. And now you see on the left the same thing that you saw before. You see the two colors on the two edges while I'm moving. And then on the right, you see the motion flow, and there is this circle. No, I should do this. There is this circle here that tells you how the color represents the direction of the motion, but of course the direction of the motion is also represented by the arrows, but they disappear quite quickly. So now I'm just going to try to show you all the colors. So you see the circle that you have on top. Now I'm just sampling it by doing this movement, and then I can do the opposite one. Yeah, and I think that's a good time to start the break.
Okay, we have a break now, and you can get a coffee, a tea at the bar nearby.
So welcome back. I hope you had a good break. So now we set the stage to talk about how insects avoid obstacles. So that's the next part of the talk. And before we go into it, I want to show you again what neuroscientists are doing in terms of experiment with insects to understand how they avoid obstacles. And this is a first movie about an experiment recently done at, in Bielefeld University by some colleagues. And this is an animation to show you how the experiment is constructed. So a bumblebee is flying into this tunnel and avoiding obstacle. And so this was just an animation of how the experiment is done. And in the next video, you will see a real bumblebee flying in that tunnel with obstacles. And then you see how they fly. They just go around, check the obstacle, and then go around it. And then slow down. No. So those are the kind of experiments that they're doing. And here there is another video about the kind of research that we do after we build a robot and we put on the robot a brain that is supposed to operate similar to the brain of the insect to achieve obstacle avoidance. And this is a video that appeared on the
experiment with bees is to put them into a tunnel, and you can see that they fly perfectly in the center of the tunnel. Now we want to do the same experiment with the robot. So it should perfectly center in the tunnel because it's seeing the opening of the tunnel at the end. We built the robot on one side to understand how collision avoidance happens in insects, and on the other side to see how well neuromorphic hardware performs. So this is the robot I was talking about, and it contains here a camera in front, and then we have here the main processing units. Collision avoidance means that when you're in a cluttered environment, that you try to avoid the obstacles surrounding you so that you never crash into an object and get hurt or damage yourself. So now we're going to put an object in the middle of the tunnel, and I put a lot of structure onto the object so that it's easy for the robot to see the object. Okay, that looks correct. These systems can then be used for autonomous navigation, such as autonomous cars or vacuum cleaner robots.
Yes, I told him, why did you finish with the vacuum cleaner? But so that was Torben Schoppe, a former PhD student of my lab. So now let's look into how do insects do and how we put it into the brain of our little robot. You might remember this picture. You're sitting on a train. The train is moving. There are some trees close to the track of the train. They're going to pass in front of your eyes very fast. But the houses up there and the hills, even behind the houses, you're going to see them for a long time. They're still going to move, but very slowly. So now we're talking about optic flow again, and we're talking about the fact that the intensity of the optic flow has information about the distance of objects. So the motion is caused by ego motion. Because I move, the objects are moving, they are fixed otherwise. And the closer they are to me, the faster they're going to move in my visual field. But I'm not telling you the whole story, because in fact, this is the case only if I move in a translational way. So only if I move in a straight line, then I have this very good information. Because if I'm standing still and I start to rotate, I see things moving, but they all move at the velocity of my rotation. That's what the picture up there is showing you, that if you're doing a rotation and you look at the motion flow represented by this arrow, they all have the same length, which means the motion flow, the optic flow, is the same everywhere, because it's given by my velocity while I'm turning. But if I instead move in a line, the motion flow, so the length of this arrow, the intensity of the optic flow, tells me that those things are close, but those are far away. And it is very well understood that insects use this information to avoid obstacles. But how would you separate these two motions if you're going to move in any arbitrary way? This is something that is possible, but is computationally expensive. And insects don't have so many resources. So a fruit fly has 600,000 neurons, a bee has about 1 million, but they need those neurons to do many things, not only the motion flow perception and computation. So how do they solve this problem? You have heard in the first video, I believe, about saccadic flight. I don't think the video explained it very well, but saccadic flight is about the insect flying straight and then making a turn. And then they fly straight, and then they make a turn. And there is a beautiful picture here. I should hold my pointer so that I can switch. And so that's saccadic. Oh, but I was told I shouldn't move too much, so I go back here. So that's a beautiful picture of this maneuver that a fruit fly is able to do, that really shows you this saccadic fly thing. So to go back, they don't have the computational power to disentangle the two motion flow if they move in an arbitrary way. So they solve it by behavior. Their brain is limited. They cannot do the computation. So they solve it by having phases in which there is only translational motion. And then in those phases, they can perceive the distance of objects, and then they can decide which is a safe direction to turn to. So we started from this knowledge and from our knowledge of other computational primitives that we know exist in the brain to build a model of obstacle avoidance in insects. And this is a complicated diagram with a lot of boxes, but I will guide you through it. So the first big box here is implementing something that we call spiking elementary motion detector. So the elementary motion detector is a very well-known model, also called Reichardt detector, that many years ago was proposed to explain how insects perceive motion. And we did it in a novel way. So this box here tells you how we constructed our elementary motion detector. We use an event-based camera, what you have seen already. We do some filtering. This is spatio-temporal, I don't remember. It's a long time ago. I don't remember this acronym, but I know what it is. So what we do is, you have seen that there is noise in these cameras. If nothing moves, you still see this activity. And so we want to make sure that we compute data that has a meaning. We don't want to try to figure out something from the noise. So what we do, instead of using every pixel, we take a 2x2 square pixel, and we integrate over time. So that's why there should be there an S for space, for the 2x2, and a T for time, for the integration over time. So by doing this, I make sure that I see something only when there is really an object moving, and not just because of noise. So if an object is going to pass by my 2x2 pixel, they will be activated more or less at the same time, so I'm going to see activity there. And then I do a similar construction to what you have seen before in the second demo. But now, because I have a roving robot, I'm not flying, I don't really care about all direction, I make it as simple as possible. So I want to see right to left and left to right. So that's why I have two populations of time difference encoder, left, right, right, left. And then again, because I'm not flying, I cannot decide my altitude, but you have seen also in the videos that the bumblebee doesn't really do this. They fly at the same altitude, and then they're avoiding the obstacle. So I also, instead of caring of where the motion is in terms of at what height, I collapse everything in one line. So that's what I see here. This is an integrator where I collapse all my visual field in one line. So now I have one vector. And at each location in this vector, I have activities of my neurons. And the more the neurons are active, the more motion is perceived in that region of the visual field. So now I have a robot that is moving. And in this vector, I have a representation of the total optic flow at different position in the visual field. And now, basically, what I want to do, because I want my robot to be safe, and I know that safe is where little motion flow is perceived. Because if I have high motion flow, means that things are close to me. So I don't want to go in that direction because I'm gonna end up in a collision. So I look at this array of activity, and I want to select the region of the space with less activity. And so this operation, which is in this case inverted, it's something that we call winner-take-all. So that's the WTA you see here. It is inverted because the winner-take-all normally is going to select the region with the highest activity, but now I want to select the one with the lowest activity. So what I do is I provide some noise activation to the winner-take-all, I make it active, and then the motion flow is going to suppress activation. So that's why this arrow here, it's labeled as inhibition. So this is suppressing activation. And so the winner-take-all is going to then select the region of space with the lowest motion flow. There might be a situation where motion flow is too high everywhere. My environment is very crowded. I'm moving in a street, and there are a lot of, for example, I'm a car, and for some crazy reason, there are a lot of cars coming in my direction. So I probably don't want to go anywhere there, and I have to figure out what to do. And this acronym, I remember it, ET, it's escape turn. So I'm like, I'm going straight, but everything looks crazy here. So what we did is we put some neurons here that are also part of this winner-take-all. And so if there is too much motion, those neurons are going to win. And then the robot does this, and then it goes straight again, and it tries to see if there is anything safe to go. The outcome of this winner-take-all, of this selection of the region in space with less optic flow, is going to directly drive my motors. So the robot goes straight, decides where is a good direction to go, and then turns in that direction. And then it will go straight again. So there is another ingredient here, which is the total optic flow. So if you remember well, this was the array with all the optic flow. And now there is another thing that we added later, because at the beginning we just thought, okay, when we go straight, we have a constant velocity. We are doing things simple. Let's put a constant velocity. But I guess you all know that when you are biking in the city, and yeah, for me it was kind of a panic situation. I'm not Dutch. I could bike, but I didn't know that here you need extra skills. So especially in summer when the boats are going through the canal, eventually they have to open the bridge so that the boat can go through. And then all the bikes that arrive at the bridge, they don't stay on the right because, yeah, why? It's going to be a long queue. So they're everywhere on my side, and then they're everywhere on the other side. And then the boat is gone, and the bridge closes again, and then I have to go. And I'm going to go very slowly. I'm not going to go at the same velocity I was going before. So we adapt our velocity depending on how many obstacles we have in front of us. So it was a very stupid decision to put a constant velocity. So eventually what we did is that we also have something to figure out, how many bad things were there in my environment. So I can take this vector of optic flow, and I can just sum up everything together. And so if there are a lot of objects, the sum will be very high. And then I use the value of the sum to modulate the velocity. So if the sum is very high, I reduce the velocity. If the sum is low, I let the robot drive faster. I think that's everything. I have a more complicated diagram block in case you're interested later on, but then it will be even more challenging to remember all the acronyms. So we put this idea of a neural network that can do obstacle avoidance, and this is like our hypothesis for what the insect brain is doing. So our hypothesis is that the insect brain is looking for the region in space with less optic flow. So we use a camera of the same kind of this one, not exactly those ones. And then we use the Spinnaker system, which is a platform for simulating spiking neurons, exactly what we need to implement our model. And to give you a sense of the size of the model, we use 4,000 neurons and about 400,000 synapses for our model. Again, the neurons are doing the integration, and the synapses are the connections among the neurons. Then we built the robot, and we tested it. This is similar to what you have seen already, but this wasn't the video for the crowd. This was really running the experiment. We had a camera from the top that could track the position of the robot, and we wanted to characterize the behavior of the robot such that we could compare the audio here is not really relevant. We could compare with insect behavior. And here you see that it's a common question. You have this high contrast pattern. If you notice, they are of the same kind that the neuroscientists use in their experiment. It was visible in the first video. And of course, it makes the perception of the camera just sharper. But then the question is, does it function also in a normal environment? That's why we added this part of the video where the robot is going around in a more natural environment. And then we characterize the behavior of the robots. For example, we look at things, okay, what happens if I vary the width of the corridor, the width of the tunnel? And we make observations that are very similar to what we see in biology, which is that if the tunnel is very narrow, the robot is really going to go straight because it must center itself in the corridor to make sure that it doesn't collide with the walls. But if there is a bigger corridor, you'll start see a bit more oscillation in the trajectory of the robot. Doing this kind of experiments and this kind of measurements is really time expensive. So it is quite common to simulate robotic experiment in a simulated environment. Unfortunately, often these simulations are only a simulation and there is no real robot, and then you end up having a problem which is called reality gap, because in the model you are not able to put all things that are in the real world. So for us, it was important to first see that things could operate on the robot, on a physical robot, but then we were anyway forced to move to a simulation, because there you can do much more and you can get much more data in a shorter time. So, for example, we look at something like a forest for the insects, so something that we call a cluttered environment. So this is now, let me get the pointer, this is now a simulated arena for the robot, where you see obstacle represented by these blue squares, and the red trajectory is the motion of the robot. In this particular experiment, the robot doesn't have any goal. It doesn't want to reach any particular place, and it's just randomly exploring the space. So that's what it does if there are not so many obstacles. It just wanders around and explores the space. Now, if you increase the number of obstacles, the robot cannot really go everywhere, and it's gonna basically find corridors. So it's gonna find spaces where it can navigate safely. And then, like here, if he sees something that looks like, okay, there is nowhere to go, it will do an escape turn and then go back into the corridor. But keep in mind that, you know, we don't give the robot any high-level instruction on what to do or where to go or what to look for, except this very simple strategy of look for the region in your visual field with less optic flow. And then if there are way too many objects, the robot is trapped. The robot has a sense of its body size, which is when I told you about the winner-take-all and the fact that from the integrated optic flow to the winner-take-all, we send inhibition. So you can send inhibition to the position, to the same position. Both the integrator and the winner-take-all, they are representing the position in the visual space. So you can send from the integrator inhibition only to one neuron. So every neuron inhibits the neuron in the same position. But you can also decide, okay, I inhibit this neuron, and I also inhibit this too. And you are forced to do this if your representation is such that this angle represents your body size. You know, if your body size is bigger than one neuron, that the angle represented by one neuron, you have to inhibit also next to it. Otherwise, the robot will try to enter a space that is smaller than its body size. So by using this lateral inhibition, we can basically tell the robot that, you know, this space here, for example, wasn't suitable. But we don't tell it in a high-level way. We just tell it by encoding the spread of the lateral inhibition. Then we can quantify these results. So in this kind of experiment, we have three different situations represented by these three experiments. So this is a situation where the robot can wander a lot, and so we don't want to run this simulation forever. So we decided to have a time over. So if the robot is happily going around and it never goes outside of the arena, eventually we stop the simulation. And this is a success run. So the robot never had a collision. So this is a success run in which we stop the simulation. Also here, we stop the simulation, but we stop it because the robot went outside. And here, we also stop it after some time because it seems like that the robot is doing fine. But here, it could also be that the robot is going to collide with an obstacle. And so that's the third situation in which we stop the simulation. So that's what you see here. We are characterizing the performance of the robot by plotting here as a function of the obstacle density on the x-axis. So this is how many obstacles I have in my arena. And then I have the three outcomes of the experiment. The red circles are collisions. The blue squares are the situation in which the robot left the arena. And the purple triangle are, I stopped the simulation because time was over, but this is a successful run. So what you can see is that if there are a few objects, the robot is always successful. So there are no red circles here in this region. It's always successful. Sometimes time is over. Sometimes he leaves the arena. Then when you increase the number of obstacles, you start seeing failures. So those are the red circles. And those failures, they increase in number the more you increase the number of obstacles. And so this is what is represented in the plot below. Again, you have obstacle density on the x-axis, and now you have success rate on the y-axis. So for example, if you look at this area, here you have success rate of 100% because every experiment that I ran was a success. The robot didn't make a collision. But here I start seeing that the success rate goes below 100% because I have one failure here and I have more failures here. And so now I'm also comparing two situations. So I'm looking at the success rate in case of a fixed velocity, that's this plot here, and the success rate in case of the adaptive velocity. That's what I explained to you before. It's really not a good choice to have a fixed velocity. And with this experiment, we also showed that for this system, it is a good solution to make the velocity dependent on the amount of obstacles visible. So you see that the robot can recover some of its performance thanks to the adaptive velocity. Then we look at an experiment that is also often performed with insects. Again, there is an arena. In the middle of the arena, there is a wall with two openings. And you can vary the size of one of the two openings. So now, on the x-axis of this plot, okay, sorry, here you see the trajectory of the robot. The robot is just wandering around, again, just using our algorithm. And we just look at how many times the robot is entering one of the two openings. And we repeat the same experiment by keeping one opening fixed and by varying the size of the other opening. So here you see the gap size, the opening size of the one that we vary experiment by experiment, while the other one is fixed at 10. So first thing to observe is if the two openings are of the same size, the robot does 50-50. So they are the same size. The robot goes 50% of the time through one and 50% of the time through the other. And this size is already big enough that if I then make one of them bigger, it doesn't really make a difference because they are both very big. So it keeps on doing 50-50. But if I look at the probability of entering a much smaller gap size, so for example here, the probability of entering the gap size of 5 with respect to the one of 10, it's between 10 and 20%. So if one gap is really small, the robot will avoid it as much as possible. And there is another interesting effect here, is that we also have the two colors here that are encoding the size of the arena. And what you see is that by making the arena smaller, you have a higher probability of entering the small gap. So the smaller the space available to the robot, the more it's gonna will to enter a small gap. And again, very similar behaviors are observed in the biological counterpart of our model. So if you do this experiment in insect, you're gonna see very similar behaviors. And so most of these experiments were run to kind of confirm that our model makes some sense. So we didn't instruct any behavior in the robot, and we see this behavior emerge, and the behavior looks like the behavior of the insect. So this is an indication that our hypothesis kind of makes sense. Of course, it's no confirmation that this is 100% how the insect does it, but it tells us that it is a reasonable hypothesis. And once we get there, then we can start asking questions. We can start doing experiments that maybe haven't been done in the biological insect, to come up with hypotheses that hopefully we can tell then the neuroscientists such that they can do those experiments. So this is the going back and forth that Carver was talking about. So here is an example of such an experiment. So here we have a corridor that becomes narrower and narrower. And I told you that this lateral inhibition, I have to make sure is correct with respect to the body size. So I have to make sure that it represents body size. But then I can also think, okay, I have to have the minimum lateral inhibition such that I'm safe. But then I can also decide to make it wider. I can also decide that in some situation, I want to be extra safe. I'm not going to go through a gap, even if it's slightly bigger than my body, because I don't need to. So the thinking here is, there is an insect. At the end of the tunnel, there is food. And the insect just ate food, or the insect hasn't been eating for 3 days. So now the black insect is the one who's really looking for food. And what we did to our virtual insect, to our robot, is to limit the spread of the lateral inhibition. So this 0.0 means that I'm only taking care of body size, and I would take the risk. So I'm saying it could be that an insect could modulate its willingness to take a risk depending on its state. And what you see on the other two trajectory is the situation in which we spread a little bit the lateral inhibition and then even further. And you see that automatically the behavior of the robot is very different. So in one case, it would go a little bit inside the tunnel, but it will stop quite quickly and go back. And in another case, that's the insect that just had a lot of food. He really doesn't care about going for food, and he wants to stay safe, so he doesn't take any risk. So it's time to draw some conclusions. I was trying to convince you that a complex system with animal-alike behavior can emerge by combining local neural units in a bio-inspired architecture. So as I already said a few times, we didn't program any particular high-level behavior. We didn't tell the robot what to do in the corridor, what to do in the cluttered environment. We only put inside the brain of the robot a model of what we think is happening in the biological brain, but at a very low level, which is find the region in space with very low optic flow. We implemented this architecture in a distributed, parallel, and asynchronous neural system, robustly performing biologically relevant tasks in closed-loop experiment, end-to-end spiking, neuromorphic from the sensing to the motor command. So the last part is more for the specialists in the field, but before this robot, I think there was no system that was using spikes, these events that we talked about at the very beginning, from the sensor to the actuation, so deciding how to steer the wheels, in a totally autonomous closed-loop system that could operate using these biologically inspired principles. I read it first and then I explain you what I mean by it. Once hyperparameters are tuned to achieve desired behaviors of local neural units, different tasks can be performed in a variety of environments with the same single set of parameters. Identification of robust computational principles. So what we mean here is that often when you build a model of this kind, you have many parameters that you have to set. You have to decide the time constant of the integration. You have to decide the amplitude of the currents, as you have seen in the first slides. So those are all parameters that you have to put in the system, and there are many. And then you might find a set of parameters that make the system work in one condition, for example, in the tunnel. But then if you move your system in a cluttered environment, it will stop working. If that's the case, probably your model is not very robust. So that's what we mean by identification of robust computational principles. So going back again, the principle is avoid high optic flow. That's the principle. We set the parameter to realize that and only that. And then we don't change them again. And we place our robot in completely different environments. And in all of these environments, we observe behavior that make a lot of sense and also resemble the behavior of the flying insects. So that's the claim we want to make. The claim is that this research leads to reasonable hypotheses and prediction that can be validated with neuroscience experiments. So we are trying to close the loop from going from our understanding of the neural system, building an artificial system and using the artificial system to guide research to better understand the biological system. And I want to leave you with one question, because one reason why I decided to study physics is because I could learn things by understanding. Not much memory was needed, but what was needed is an effort to understand things. And once I understood something, I would remember it. So I want you to remember something about this talk. And basically, I want to change your brain forever, because I also study learning. So I know that if you will remember something, it means that some synapses in your brain changed. So I want you to figure out something out of this talk, which is the answer to this question that I'm not going to answer myself. Why do flies and other insects beat themselves to death against the inside of windows? And I hope that by using the knowledge I tried to provide you today, you can reason about what could be your hypothesis of the answer to this question. And it might happen that from today on, whenever you see a fly, going over the window again and again. You might remember this talk, who knows? So, of course, I would like to thank all the people that made this amazing research possible. The two main persons are Torben Schope, that you saw in the video, and Hugh Greatorex, that is sitting in this room, but he wants to stay anonymous. And, of course, everybody else involved in the work. So those are all the people that were involved in the work about obstacle avoidance, and in general, all the people in the group. Of course, this is not possible if we don't have money to do the research, so there are a lot of funding mentioned there. And before I thank you for your attention, I want to steal 2 seconds from you by letting you know that we recently established a fund called Shruti Giri Fund. And I'm not going to tell you much because I don't want to steal too much of your time, except that this fund is going to support minority in STEM. And so I hope you can scan the QR code and find more information on the freshly made webpage. And of course, thank you for your attention.
Thank you very much, Elisabetta. We have now time for questions. Who is the happy first here?
Hi. Thanks a lot for a very nice talk.
Thank you.
I noticed in your plot, when you're looking at the success rate compared to the object density, that for both conditions you have a dip, and then at some point it goes up again. Can you elaborate a little bit on what you think about this?
Yeah, I've asked myself this question many times, and I don't have an answer. The position of the dip is different. It could be related to, from here to here, an intermediate situation in which the escape turn is not really working, because it is crowded, but it's not so crowded. So the robot is trying to find its way, and then it makes an escape turn, and then right in front of it, there is another obstacle. But now I'm making the hypothesis that this happens more often in this situation in between, and that I didn't really check. Yeah, but it could be a possible explanation. Yeah. And what we do know is that most of the failures are in that kind of situation. Most of the failures are like, where things are really complicated, and I try to run away, and then, I do a random turn, and so I'm just hoping that then I can start again, and there is nothing there, or there is nothing there close such that I have no hope anymore, and I just collide with it. But very good question.
I have a question here, Valerio.
On the diagram of the root.
Wait.
Diagram of the route, where the robot goes through the 2 openings, why doesn't the robot go up and down? Why does it want to go through the 2? Yeah, I would go just up and down, up and down, up and down, and it's fine. It's much more risky going every time through these 2 gaps.
But there is the wall. So it goes through the gap, and then everything is safe, so there is a lot of empty space, so it's continuing in this direction. But then it sees the wall here.
Why does it go up and down?
Here, you mean?
Why it doesn't always go in a line? No?
Oh, like this. So there... So you could set up the robot with an initial condition such that it will do it. So if you start the robot here, facing up, it will do that, yes. But we wanted to force the robot to use the gaps, So it is starting by looking at the middle wall, and it's making the first decision. And once it's in that loop, then the wall is sending it back. But yeah, very good question as well. So the most challenging part of this kind of talk with a mixed public and not a technical one is that you always get questions that we have never heard before, because your colleagues have seen this all the time, and so they all ask boring questions. So this part is very exciting.
Please.
So maybe I can ask the boring question.
No, please.
You've been talking about avoiding static objects, and then you have this optical flow. But of course, in reality, many objects will not be static. You must have answered this question 1,000 times.
Yes, this is a problem, especially if the other moving object moves exactly like me, the camera doesn't see it. Yeah, but chances are quite low that this thing happened for long enough, and the real world is kind of messy, and I'm gonna be fine most of the time. But we have done these experiments in a very safe way because there were no moving objects. So this is, if you want another field of research that always makes me think about another insect, which is the dragonfly. Dragonflies are very, very good at catching prey while they fly. And so they're moving, and the prey is moving, and they can make very good prediction of the position of the prey in the near future, and they go for it and they catch it. So this is also an insect that is very well studied, and those kind of studies will look at that problem that you are talking about, but we didn't consider that in these studies.
I had two questions, one here and one more in the center.
So it wasn't boring,
There
but
are
I
a lot of
was very well
questions.
prepared.
Thank you very much for a very nice presentation. But I'm trying to get more close to biology, because this is pretty far away from biology.
Yeah.
That an insect has an eye with a lot of facets.
Yeah.
Can I compare for the optic flow the facilitating and the triggering squares as being different facets in the insect eye?
So I have a backup slide for you. And this comes from the fact that I was doing this research with a neuroscientist, Martin Egelav, one of the authors of the paper, who's doing research with bumblebees, for example. So this video with the bumblebees is from his lab. And even without thinking about the facets, if you look at the insect visual system, they don't produce spikes. So the receptor, they don't produce these events, but they produce so-called graded potentials. So their encoding is analog. It's not done with these spikes. I know I'm not answering your question, but then we'll get there later. So my colleague was totally against using these cameras, because these cameras are not a good representation of the visual system of the fly, because they operate with spikes. And then the question is, how in detail you need to model the biological system to get something out of your model. And you have to find a compromise somewhere. I'm not able to build a fly, and I let my neuroscientist colleague to study the real fly. I'm not going to build exactly a fly. I'm trying to understand high-level principles. And so I made a big box that contains all these small lenses, and I assumed, they don't play a big role here, except, that of course, in the optic flow encoding of the flight, the signal is coming from different facets in the eye. And this slide is here to tell you that in the insect brain, you know, if you look at the visual system, and then you look, this is a schematic representation of what they think is the neural network in the insect brain, and these are the cells in the brain itself. And they tell you there are cells that are called T4 and T5, and those cells, they represent optic flow in the 4 cardinal direction. So exactly the way that we have done it in the model that you saw in the second demo. So what I'm doing here is, I'm not saying I'm implementing all the details. I have to abstract away from the detail. And then we also do measurements. So this is the biological response of the T4, T5 cells. And then we did measurements from our spiking elementary motion detector to see that we can observe a similar behavior. So basically what I'm saying is that in this model, when I have this big block here with those components, I'm only saying that given a visual input, the output of this box is something that I can find in the biological system after the T4, T5 cells, but also in the biological system, it's made of many blocks. And the first block is the one you were talking about. But I abstracted away. The small blocks in my big blocks here are totally different from the biology. So I'm not modeling them at that level of detail, but I believe I'm producing a model that can still be used to learn something about biology.
So with these words, I know there are many other questions, but my superiors tell me that we should stop here. And I would like to do 2 things. First, thanking 2 people. Well, first, Elisabetta for the nice lecture. We give one of the books that KNG has been producing.
Thank you.
And I would like to thank also another important person. As an experimental physicist, we always value technicians. I would like to thank the... And now I would like to give the word to Marianne.
Maria, thank you, Elisabetta, and sorry for being the one who has to say, okay, now we have to stop, because we do offer you drinks in the newsroom to continue this really nice discussion. But I also need to point out our upcoming events, because they are kind of special. So if you would look at that side of the room, you see our regular program with the Bernoulli lecture coming up, which is, yeah, but I'm pointing there because I thought, so the Bernoulli lecture is focused around mathematics. You're all invited, but you can also see our founder, Theodorus van Swinderen. And he's always looking at us like, stay on time and make sure you have a very good program. But below the Bernoulli lecture, you can see that in April, we have our Lustrum Week. So we are celebrating our 225th anniversary. And we really like to invite all of you, members, non-members, to join us for the celebrations we are organizing there. And for that, you can take a look to this side, where Theodorus is really happy to celebrate our 225th anniversary. And I would like to thank the guys from the technical assistants for doing that, because I really think he's like this grumpy old man. I think we should keep on using this one. Okay, without taking too much of your time, I need to point out that the Lustrum, so the celebrations already took place starting January with a lot of really nice movies, which many of you attended, but if not, there's still the Ammonite movie on Sunday, March 22nd, to join. You can sign up using Studium Generale. It's only €5, and well, be on time. It's really quickly sold out. The same goes for the Kaptein lecture, which we organize within the week of the Lustrum. Tickets are already getting very, very quickly. It's for free. Again, you need to sign up with Studium Generale. And so please do if you're interested in that. And I can highly advise you to go there. It's by a Nobel laureate on ancient DNA, and it's going to be very impressive. And there was a third I need to point out, and that is the theater play. I don't remember the English translation, but in Dutch it's about goudmerels. That's the title. You can read all about it on our website. Please go there, check out all the other details, because we do organize quite a lot. But in view of time, I will not mention them all. But please sign up for these 3 events, which I just mentioned, and see you then. Thank you. See you in newsroom as well to continue the discussions. Did I now miss something there? Sorry. Yeah. So after that we have Tessa, and then after that we have the last regular lecture of this year, which will be Odie Sibon, telling about her exciting research as well. Okay, I think that was it. So thank you very much. Thank you, Elisabetta. Really nice. And thank you, Maria. And now drinks. Yeah.